So, Cocconeis and its mysteries. So lately I'm focused on freshwater Cocconeis taxonomy because definitely that is a problematic and very interesting topic. So the roadmap for today, I'm going to cover several topics. First, why study freshwater Cocconeis, the current state of Cocconeis placentula species complex taxonomy. My work in presentation is focused on Cocconeis placentula species complex. Then a brief overview of Cocconeis frustule. I'm introducing here the terminology that I use and detecting structural elements and all challenges associated with that. Then I'll talk about the integrative Cocconeis taxonomic studies and will present SEM, scanning electron microscopy description of 7 Cocconeis species from streams in California. And we have DNA for 5 of these species currently. And then I'll make comments on the diagnostic value of Cocconeis girdle. And then I will finish with a practical identification guide. At this point, mostly suggestions for taxonomically important characteristics and next steps of this research. So why study Cocconeis? Especially in streams of California, which are mostly alkaline, Cocconeis is a very common genus. So I'm working with streams in California for 15 years. Cocconeis is almost in at least 80% of the samples. I see it attached on Cladophora, most commonly attached on Ulva, which is a little bit more unusual, Vaucheria. And in Northern California, Cocconeis valves are visible on mosses and very interesting green alga Proseola, which is macroalga. And what is most intriguing to me, I'm seeing Cocconeis also attached to other diatoms, like the chain-forming Lerbekia, Pleurozyra, even on Eunotia, which is really strange, and sometimes on Melozyra. Also, there are endemic Cocconeis species described from the Pacific Northwest, more specifically the Cascades Mountains, which are obviously hotspot of Cocconeis diversity. Here you see the rape valves of 2 endemic species to North Pacific Northwest, Cocconeis clamatensis and particularly Cocconeis rugosa. Maybe clamatensis, I mean, their records is maybe a little bit more widely distributed. And these 2 species are described from area which is on the boundary between Oregon and California. And I've seen both species, of course, very rare, with a few single valves in some samples in Northern California, which really triggered my interest and indicated the fact that probably Cocconeis has a quite good potential to be indicative at different ecoregions, because the species that I see in Northern California are quite different than the Cocconeis flora in Southern California. What is the current state of Cocconeis taxonomy, particularly Cocconeis placentula? We have quite significant difficulties identifying distinguishing species within this species complex. We quite often identify that some very wide and unclear taxonomical resolution. And currently in diatoms.org, which is our main reference when we identify species diatoms from North America, Cocconeis placentula is presented as complex, and 2 varieties, Euglypta and Lineata, still not included in the website, and Pseudolineata. And here I directly refer to the opinion, which is the current view on diatoms.org. There is lack of clearly established differentiating criteria between this taxa within Cocconeis placentula complex in the biomonitoring programs. Neither original descriptions of this taxa nor the descriptions available in the floras are sufficient to separate species at any degree of certainty. And that is really what we see when we analyze samples with freshwater Cocconeis. We see all this incredible morphological diversity and variation, and we have difficulties to separate species. And here, Dr. Yan and her team did a great start of studying Cocconeis by establishing and leptotypifying the complex of Cocconeis placentula, starting with the nominate variety, Cocconeis placentula, and also leptotypification of Cocconeis lineata and Cocconeis euglypta. The information in this case is coming from monoclonal cultures, which are sequenced, and there are nice and clear descriptions of this taxa, which are a great start, and it's building system that we could continue developing and growing. But still further studies are necessary to determine whether and how individual species within this complex may be distinguished using morphological characters. And this is where my work is starting. Here I'm going to introduce a little bit terminology. All we know that Cocconeis is a heterovalval species. It has two different frustules, the rave valve, and I'm introducing the abbreviations that I'm going to use along of the presentation. So the rave valve is abbreviated with RV, and also the sternum valve, which is missing the rave, of course. And most importantly, the entire frustule of Cocconeis has a quite complex structure. The frustules of Cocconeis placentula complex are usually, they're nearly straight, and the girdle is so narrow that usually we see the frustule from the valve view, either a rave valve or sternum valve. And because of this reason, usually we characterize and identify Cocconeis either from rave or sternum valves. We use characteristics like the position of the areolae. Of course, we are starting with the size and shape and outline. And also we use the areolae on the, okay, so here we are measuring the width and length, of course. And then we are measuring the areolae in 10 microns. This is exactly how I want to show here clearly how I'm measuring the areolae in my studies. I measure them in 10 microns exactly in the middle of the hemivalve in the center. And then additionally, I measure them close to the periphery, where usually they are much denser, and close to the central area. Also, when we look at the sternum valves, it's important to see how many longitudinal rows of areolae are present, and we are counting them within the entire half of the valve. We are counting the number of these rows of areolae and how they are distributed, what is their pattern. This is the traditional approach to Cocconeis taxonomy, and here where the confusions are coming because the variation is so high and we have difficulties splitting species. Of course, the same measurements are done in the rave valve, and for the rave valve, we pay attention to the submarginal hyaline ring, which is kind of breaking up the areolae and the striae. And also, usually we count the number of these submarginal areolae. Lately, that was mostly suggested by Dr. Yan, we started looking with much more attention at the apices, because these submarginal areolae, they may be continuing and surrounding the end of the rave, or there may be a gap And we see that tendency that it could be taxonomically important. And also we pay attention of the distribution of the areola on sternum valve, specifically at the apices. Then, of course, when we check the ultrastructural details, we're looking at the areolae. They're covered by very thin hymens, which are lattice-like, and they have these very tiny slits. Here to the right, I'm presenting the areolae openings on the sternum valve. As you could see, at least to me, I don't see any variation of the occlusions of this areolae, and because of that, I think that they are not taxonomically important. If we look at the occlusions of the rave valve areolae, which are, you see more roundish, here we may see a little bit variation. So the hymen is visible from the internal valve view, and we are noticing here some minor variation, which may be taxonomically important. And sometimes the external view also could be a little bit variable and specific for different species. And now we're looking at the girdle view, which is typically it doesn't really get a lot of attention in our studies because it's really hard to be observed. The girdle of Cocconeis is composed of coccolae. These are very thin connecting bands of silica. And let's look a little bit more careful here at the girdle in this entire frustule. I think that it's clearly visible that there are three bands or three copulae. In the central one here, which is indicating, so in the middle, it's not connected to any of the valves. But the very important, taxonomically important bands or copulae are this one, which are connected to the valves itself, and they're called valvocopulae. And I'm going to talk a lot about that. So there is one valvocopula which is attached to the rave valve. And the abbreviation that I'm going to use here is RVVC, which is the rave valve valvocopula. And then there is a second one, valvocopula, which is attached to the sternum valve, and the abbreviation here is SV valvocopula. Looking at this picture here to the right, we start looking internally, what is happening inside, I mean, how the valves, they look from internal valve view. We have to make difference between the mantle. The mantle is the very last, that is the end of the valve, which is turned down. And in fact, the valvocopulae are closely attached to that mantle. Also, one structure which is really very rare to observe, I mean, I have only these two microscopy images. The coccolae, they're connected by this very tiny element, which is called ligula. It's really hard to see in the slides or with electron microscopy, so we don't have enough information on this one for Cocconeis placentula. So our current knowledge on the structures of the cingulum in Cocconeis is based on this article from 1982 by Holmes, Crawford, and Round. What we know about the cingulum in Cocconeis placentula complex is based on two species indicated here, placentula and a species from Northern California, quite large. In fact, it's not identified, but it's listed as CF roxy from stream in Northern California. We know that the cingulum of each valve consists of one valvocopula and two additional bands, but these bands are developing only toward the end of the life cycle, just preceding the cell division, so they are not always visible. Also, we know that the rave valve valvocopulae is less robust than the sternum valve valvocopulae, and they have different profiles. The rave valve VC, so from now on, I start using this abbreviation VC, is fimbriate. It has these extensions on or fimbriae, which in most species, the ray valve VC has this fimbriae, and they're variable, and they're diagnostic of each taxon. Also, we know that the ray valve VC in Cocconeis placentula is open. That is also true for the second species illustrated here. And there is not that much information on the sternum valve VC. We only know that the sternum valve usually is not developing this type of fimbriae, but still it has a rough surface which is in contact with the valve, or sometimes it could be denticulate. No illustrations of the sternum valve VC in that article, which is giving the impression that it's not important. I obtain a lot of electron microscopy images of Cocconeis to be able to understand completely the ultrastructure of this genus. And I could say that the interpretation of the valvocopulae really could be confusing in some situations. And confusions also exist in the literature. So let's look at the ray valve of Cocconeis placentula. The images here are coming from a monoclonal culture, which is sequenced from our studies in California. So we clearly see here that the ray valve VC, valvocopulae, it's open, it's attached in a very specific way. The fimbriae, in fact, you see that they're nicely fitting the hyaline rim. And then the ray flace, or the sternum valve, in this case, to me, it's obvious that the valvocopulae, in this case, it's attached to the mantle. And in fact, this dark line here is indicating that is the connection line between the mantle and the valvocopulae. We see that this valvocopulae really is missing the large extended fimbriae. It has a quite different structure than the VC of the ray valve. And then how this band, which seems to be completely closed, we don't see any openings here, how it's attached to the valve. Looking at this image here of the valve and magnifying the area here, this is how we judge about the connecting elements. The valvocopula of the sternum valve is connected to the valve itself through that area, which is undulate. It has these regular undulations. But the confusion is coming because sometimes we see sternum valves, why they have this fimbriate valvocopula, what it is. And sometimes it could lead to the wrong misinterpretation of the figure or misconcept that the sternum valve probably has a fimbriate valvocopula. But if we look a little bit closer here in this area, in fact, we're going to realize that that valve has two valvocopulae on the top of each other, and that is, in fact, here the blue arrow is indicating the actual sternum valvocopula, which is covered by the ray flace valvocopula. So let's keep going here. Because of that, we need to look much more careful of sternum valves, which don't have associated valvocopula. Here you see just the edge of the mantle. And here we see the same valves, but they have a close, smooth valvocopula, which is attached to the edge. I hope that these two images are really different in view. And here, with a little bit more close look up, we see how the smooth, so-called smooth sternum valvocopulae are attached to the valve. They are attached by these undulating structures, which really tightly adhere to the internal surface of the valve. And again, this kind of broken valves are really valuable because we see the connecting elements. We see here the sternum valvocopula and the reflex valvocopula on the top of that. Another misinterpretations that may arise or difficulties, for instance, that is the sternum valve of new species to science, which will be soon described. We are working on this manuscript. And again, what we see here, we see the sternum valve without any valvocopula. We clearly see the age of the mantle, then the same species with sternum valvocopula closed and smooth, which is attached to the mantle. And again, similar valve, but in this case, the valvocopula looks differently, and it's open at one end. Why is that? Because in this case, in fact, again, we observed both valvocopulae on the top of each other. So my point here is that we really have difficulties even reading the images, and we have to pay much more attention, even if we have a really detailed and nice visualization of these structures. It takes a little bit time and processing of the information, what exactly we see and how we describe it. We would prefer to work with cultures, with Cocconeis. Because all we are dealing with environmental samples, we know that usually the environmental samples contain mixed populations of Cocconeis, which cannot resolve questions like overlap between species, morphological variation within species, and we cannot establish the species boundaries. And because of that, our preference, if we have this option, is to work with cultures, with monoclonal cultures of Cocconeis, which we call integrative studies, because they provide us with the ability for DNA extraction and genetic information about the species, but also it provides us with a very rich material for detailed ultrastructural observations, and it allows us to be very confident when we are describing species. We are able to link all complex components of their fructules with really high confidence and precisely to observe and describe their ultrastructure and to apply this knowledge to phylogenetic species concepts. And I was extremely lucky that Dr. Regina Yan and her group in Botanical Garden and Botanical Museum in Berlin were so willing to work together on Cocconeis taxonomy from streams in California. In general, I'm working with, I'm culturing algae, I'm culturing quite successfully cyanobacteria. I try to isolate Cocconeis. Cocconeis is really very picky and very hard to work in culture. I was not able to do it. And because of that, I'm extremely thankful to my German colleagues that were interested to do that study together and to isolate so many Cocconeis streams from California to be able to apply integrative taxonomy approach to the species. We started with one culture, provided, isolated Cocconeis culture isolated in 1982, 38 years ago, and deposited in the Utex algal collection, which was identified as Cocconeis placentula variety lineata. But the sequencing of this strain didn't really fit the system, the already established system and gene data for Cocconeis lineata. And that was our first study with Dr. Regina Yan and Demetrio Mora. So I had access to this culture, I ordered it, and it was my first experience working with Cocconeis cultures, which showed me the incredible opportunities that we have when we study Cocconeis from unialgal culture. We have been able to describe this as a new species to science, but particularly to me, that really shows the huge potential of using monoclonal Cocconeis strains for detailed observations of their ultrastructure and very really deep and hopefully correct descriptions of their morphology, particularly specifically related to the structure of the connecting elements. And since then, we did a study of samples collected from California in 2021. With the help of many colleagues, we were able to sample 11 stream watersheds in California. 8 of these watersheds were distributed near San Diego, which is logical, right? It's very convenient. So half of these streams are urban streams, but also the streams which are more to the east are non-perennial streams with high reference status. Also, we had 2 streams in Central California, urban streams in San Francisco Bay Area, and 1 stream in Northern California, which is located in a hotspot of diversity, where new species of Cocconeis were discovered. So the fresh samples were collected overnight or as soon as possible shipped to Germany, where when 80 clonal Cocconeis strains were isolated. It's totally amazing. It's a very hard work. And these strains were sequenced. So we have gene data for the majority of the strains isolated. We have rbcL and 18.6 genes sequenced, and based on the genetic information, we were able to separate 8 species. And here I'm going to present 5 of these species because still 3 are not, the data are not completely analyzed. And also we have been able to do very detailed scanning electron microscopy of this species. So I'm going to talk about Cocconeis placentula, which was isolated from 5 sites, 2 sites in Central California and 3 urban sites in Southern California, Cocconeis lineata in 1 site, and species which is very similar to Cocconeis lineata from 2 sites. Then we have been lucky to sequence Cocconeis cascadensis from that site in Northern California, and we are in the process of describing a new species to science. I hope that it's okay to share the name of this novel species, Cocconeis ectori. That is work in progress. I hope soon to see it published. So I'm going to briefly provide overview of each of these taxa and I'm going to use the same way of organizing the electron microscopy images. Here on the first row, I'm going to show the images of rave valves along with the corresponding rave valve valvocopulae, and on the bottom row, I'm going to illustrate images of the sternum valve. So what we know for Cocconeis placentula, some characters are really stable across populations. So we have 5 different populations of Cocconeis placentula, isolated and sequenced. It's amazing, but in all 5 populations, the external sternum valve has exactly the same ornamentation. It has a very long slit-like areolae and these tiny dots, which are very fine perforations on the surface of the valves, but they don't penetrate completely to the internal side of the valve. That feature was quite consistent among all these 5 populations, which are coming from different areas in California. Also, the position of the areolae and the areolae pattern was quite consistent also among the populations, and we were very strangely surprised here to learn to get a little bit more clear information about the sternum valvocopulae in Cocconeis placentula. It turns out that in fact it's smooth and it's closed, as indicated here on this image. You see that this band is completely closed. There is no opening. Unfortunately, I don't have a very good illustration of this closed valvocopula because I didn't didn't spend a lot of time doing electron microscopy on placentula, because obviously it's not a new species and it doesn't need a detailed description. But Cocconeis placentula, really, it has a sternum valve with the closed valvocopula, which is smooth, and it's connected with the surface of the valve or the mantle with these regularly undulate structures. And more images of Cocconeis placentula. The valve outline and the size are definitely not indicative. There is a huge variation. You see here to the top, obviously this is a oxospore, and that very small cell here is nearly round. It's not elliptical anymore. So, and also there might be teratologies, as you see here to the left. So the valve outline and size is really hard to be used for the delineation between the species. And for this particular slide, I put all images at the same scale just to be able to compare sizes, which is not the case of the rest of the images. So the next species that we have in culture is Cocconeis lineata from California. The interesting, it's quite consistent with the lectotype of Cocconeis lineata. And here the information that we are providing as probably new information, looking more careful at the rave valvocopulae, particularly on this image. You see how it consists of 2 equal halves. They are not simply broken. Their ends have this very specific shape. So now, and even getting ready for this presentation, I realized that probably the rave valve in Cocconeis lineata is open on both ends, as indicated here by the red arrows. So we may want to look a little bit more into that, but I'm almost pretty sure that that is the case. And you see the challenges interpreting images like that. So here to the left, you see illustration of sternum valvocopulae. They look differently from their both sides. At the valvular side, which in fact is connected, that is the side which is connected with the valvocopulae. It has a rough surface, or in fact it has this attachment rim, which have these undulations, which are quite regular undulations, and that is the connecting point with the valve. But the other side of the valvocopulae, or the upvalvular side, which is away from the valve, it's smooth and it has a different profile. So then we have one more species, which is very close to lineata, but genetically, in fact, it's indicated here with the Cocconeis CF lineata, because genetically it's slightly different. I don't know if it's going to make it as new species, or we are just going to extend the description of Cocconeis lineata, but in general, the species is very similar with Cocconeis lineata. the only difference that I see morphologically, that the valves are a little bit coarser, a little bit larger. Maybe the areolae are a little bit better developed, but in general, as structural elements, it's again the same and very similar to Cocconeis lineata. So then the next species, which we believe it's, genetically it's very distinct, and it's new to science, and it's quite widely distributed in California. It's going to be described as new to science. What is interesting about this species is that it's morphologically kind of similar to Cocconeis placentula, in the sense that, if we have only the sternum valves, and if we compare the sternum valves of these two species, they really could be very easily confused. But the major difference here is the structures of the connecting elements. Again, the valvocopulae. The sternum valve valvocopula, which is illustrated here, is closed, and it has undulate rim. So that is quite similar to the valvocopula of Cocconeis placentula. But the rave valve valvocopula is very different. What it's missing the fimbriae. There are no fimbriae on this valve, and they are very tiny, maybe spines, or I really don't know which term to use, but it seems that this valve is connecting only with this very tiny, short spines. These are the connecting elements on the rave valve valvocopulae, which is quite significant difference with Cocconeis placentula. Now I'm including here this slide just to see how we could transfer this knowledge to actual environmental sample. That is environmental sample from Northern California, which seems to me to contain Cocconeis lineata. So let's say that small valve here is probably Cocconeis lineata, and here are the electron microscopy images. It's quite consistent with the sequenced species. But then what is the large one? Is that the same species? Is it different species? So that slide is illustrating the difficulty that we are dealing with the environmental samples, trying to identify Cocconeis. And doing a little bit more electron microscopy work on the larger specimens, I realized that in fact that is quite different species. So again, it's coming from the same sample. The larger specimens, in fact, they have quite, in addition to the larger size and of course different areolae, size and number of areolae and so on, they have a very distinct kind of qualitative difference. The connecting elements here are very, very different than the connecting elements in Cocconeis lineata. And here the ray valve valvocopula consists of very long and distinct fimbriae. And the sternum valve valvocopula is open. So in fact, both valvocopulae, they are open, and it has a denticulate irregular rim. So it seems that in this sample we have two species which are relatively similar, the small and the one species, the small and the large species. But when you look at their connecting elements, they are very clearly separated. So then I'm continuing with the species which I described in 2018 at the very beginning of my journey with Cocconeis from streams in California. Why I started with Cocconeis cascadensis? Because it has a very distinct morphology. So that is species that could be described with confidence without genetic data, because that is quite different than the rest of the Cocconeis placentula group. It differs by really very coarse areolae, which are less than 13 in 10 microns. The areola openings are really very large and distinct and very specific. And also the rave valve has a hyaline rim illustrated here with these very distinct bumps, which are not present in any other species. So when we see something like that, we definitely very quickly and clearly may separate it from the rest of the species in the sample and describe it as very different species, and in this case, new to science. And luckily, we have been able to isolate this in culture from the only sample in Northern California. And again, you see the power of integrative studies, which are providing such an incredible material for scanning electron microscopy observation. I think that that image is great because you see how material like that provides with a lot of valves. They belong to the same species. We see the changes of the size. We see everything that we need. And most importantly, we are able to connect elements and to know what is the structure of the entire frustule. And here, very quickly, the last species which I described without genetic data, but it again has a very distinct morphology, is Cocconeis fetcheriana, which has again very distinct areolae and closed, very robust sternum valve, valvocopula, which could be distinguished only with light microscopy. And now, let's summarize the data. I know that at this point you may be a little bit already confused with what is going on, but when you put together next to each other all these structures and valvocopulae, we see that it's information that could be organized. The sternum valve, valvocopulae, they could be either closed or open. And here the ornamentation is not very diverse. It could be either undulate structures, connecting structures, or it could be irregularly denticulate. Then compare with the valvocopulae of the rave valves. So here the variation is slightly different. Usually they are open at one end, but in Cocconeis lineata, they may be open at both ends. And here we observe a much larger variation of the connecting elements from different types of fimbriate through irregularly denticulate and small spines. And then let's combine everything together. And what we see unique combinations. In fact, I think the power of this study is that each combination of rave valve and sternum valve, valvocopulae is unique for the 7 species I'm presenting here in this work. And then another diagnostic feature which is obvious is the shape of the external openings of the areolae. Unfortunately, that is something is not easy. In fact, it's almost impossible to distinguish with light microscopy. But you see that in fact the areola openings are very different, and they are also diagnostic for the species. And here is our complete library. I put together all these 4 connecting elements, and what we have to look for, we have to look, in here indications, which are the features that we have to check when we are working with Cocconeis under the microscope. So I'm going to speed up here to allow time for questions. But the point here is that until now we were dealing mostly with the morphological features of the valves. And now when we add to that the structure of the valvocopulae, we are going to make much more confident identifications of the species. Can we see the valvocopulae with light microscope? Yes, we can. If we observe the valves, so for instance, this is the same valve. When we change the focal plane, we could see the valvocopulae, which stay still attached to the valve, but also we could observe them individually when they are detached in the slides. And then some suggestions for light microscopy observations. We may go here through more details, but the idea is that Cocconeis, we need to observe all 4 pieces of the frustule. It's like puzzle, which consists of 4 different elements. And first, we need to scan as many valves as possible, specifically large valves, which are much more clear to connect the puzzle, and then we could make a more confident identifications only from a valve view. What are the next steps? We have several more species to describe from California. Of course, at some point, it will be great to see the phylogeny of Cocconeis placentula complex. It will be very useful to develop light microscopy identification keys. And it's on my list as well. And also, I would like to go back to the slides. I have thousands of slides from streams in California to revisit them, to be able to establish the environmental ranges of all these species, which will be useful for the bioassessment. And now I would like to thank, all my colleagues that help with that study. My colleagues from Germany, also a lot of people from California who collected samples, and my current workplace, Potomac Environmental Research and Education Center. Thank you. Now I'm ready to answer questions.
All right. Thank you very much, Rosalina. I really appreciated a lot of that information, and especially your chart there at the end. I hope you don't mind. I took a quick screenshot of that, just so I hope you'll be publishing it somewhere, or maybe we can use that a little bit amongst ourselves. But it was a nice way to look at the different features among the different taxa.
Thank you. Hopefully, it was not confusing.
No, I just have to study it a little bit. It's great. So if anybody has a question, feel free to raise your hand or throw it in the chat. I'll just start off with one. Both Kalina and Sarah said it was great to see you. They were enjoying their presentation, but they had to leave. Sarah wanted to know, you stated that there were Cocconeis species indicative of ecoregions. She wanted to know if you are prepared to say if those species are endemic to those regions.
So you're asking a little bit hard question. I think that the question about endemic species need confirmation in a sense. As time goes and more areas are investigated and we have a better understanding about the distribution of species, then we may more confidently talk about and claim the fact that they are endemic. But I believe that, at least to my experience, I'm working for 15 years on these samples, and I could tell that some species are very limited. They have very limited distribution in Northern California. Some species are rare, some species are very common, and some are really very distinct for a very small area. Definitely, they have a different area of distribution, and some might be endemic.
We're going to have Marco ask a question here in just a second. So Marco, you'd like to turn on your camera. I have one quick, hopefully, question in the chat from Brad. He asks, do you see the valvocopulae breaking open with the processing of diatom material?
Yeah, very often, and that is additional challenge which I didn't discuss here. Quite often, the valvocopulae, they could be just mechanically broken when we process the samples, and that is additional challenge, and we need to observe a lot of material to be able really to realize which is broken, which is open, which is intact. I guess that we need to have a little bit imagination also working with these valves.
Great, thank you. Marco, great to see you. Feel free to ask your question.
Thank you, Rosalina, for this great work and talk. And my question is about Cocconeis euglypta. still, I was wondering if you are working also on this taxon. And until I was working in mainly the Alps, it seemed to work that this was one of the few species preferring siliceous substrate, because I completely share your opinion that they are more in alkaline waters on different types of carbonate rocks. But when I worked more outside the Alps in different geographic areas and landscapes, it didn't seem to hold this distinction. But it may be because other species are slipping in and we are not able to confidently identify and distinguish them. So just asking if you have some experience about Cocconeis species more confidently identified than geological substrata, which may also be related to the ecoregion topic that you mentioned. Thank you.
Thank you. That is a very meaningful question. My answer is that the answer of this question is in the future. I really hope that we'll have chance to analyze more samples. But I mean, by integrative approach, but by seeing really the power of analyzing samples with molecular and morphological observations from monoclonal cultures, I only hope to have chance to analyze more samples from California. But Regina, at this point, my understanding is that we don't have Cocconeis euglypta confirmed for California genetically, right? But we have only 11 samples. And because of that, I have hard time to answer this question. At this point, it's not confirmed genetically. I didn't observe material which is confidently Cocconeis euglypta, and because of that, I cannot really provide a meaningful answer of this question with the existing data. And
sorry, do you think some of these 11 samples come from siliceous rocks? Because they may all be from, they may all come from carbonate.
Probably not. I think it's probably not. Maybe the most northern, but I'm not sure about, probably not. Yeah, you're right, the geology of California is very different than what you are mentioning. Yeah, thank you.
But go ahead.
Yes.
I find it interesting that Rosalina found Cocconeis placentula that we lectotypified from Berlin, dirty waters in California. So that's very interesting. I mean, she added a lot of information on that placentula that we didn't. I think hers look much even prettier than ours, but they're the same. So I still don't understand. And we are currently also working on more German ones, and I'm Demetrio and I and Rosalina in the process of publishing it. It's highly complicated and not easy. And the problem that we have here in Germany is that we have so many species described already, and we're trying to find out if we're talking about the same species or not. And as Rosalina was showing, that the data that we have that have been attached to the Cocconeis is something that is unusable nowadays. So it's a lot of, does it work or does it not work? So it's quite interesting, quite challenging. But I think there will be, I mean, Rosalina also loved the last picture, but we could extend it to a double size by now of species to be found in California and Germany, but we know nothing in between.
Okay, so thank you again. So just for a quick note, all sites with Cocconeis placentula, in fact, we have 2 sites from Central California and 2 from Southern. They're coming from highly urban streams. Yeah, the samples from San Diego I collected on my own. So these are channelized streams which are running through the cities, right? And this is where we find Cocconeis placentula. I personally collected the sample, so I know what is the environment. And also in the Central California, I think the streams are the same. Yeah, I think that I got question in the chat. Are these freshwater? Yes, this I'm working only with freshwater. The presentation includes only freshwater species from running waters.
But interesting is that just wanted to say that interesting is that you didn't find any of Charnecki in the sample that we had found in the Great Lakes, right?
Yes, so Charnecki is very rare in streams in California. I think that morphologically I was able to identify probably the same species in a few streams in Northern California. And I think that they are always related to lakes. So they are kind of streams which are I think that it's more like lake species.
Matt, you have a question? And I would just like to pause for just a second. We're at our typical time of the day, and there's no reason the discussion can't continue, but I want to thank anybody who has to leave for coming and attending today. But Matt, go ahead.
Thanks. Rosalina, really interesting talk. Thank you for that. The level of morphological variation you're seeing just within California Cocconeis is fascinating. I mean, not just the differences in the valvocopulae open and closed. There seems to be some significant structural difference in the sternum valve in the cross section. That it's almost like there's some chambering going on in some species versus a very simple poros. But I'm wondering about the plastids. Are we still seeing sort of the same kind of C-shaped plastids in all of these? Or are we seeing some variation there as well?
Okay, Matt, thank you for this question. So just I would like to clarify about the areolae. In all the species that I'm investigated, they definitely belong to placentula complex in the sense that they are not chambers, right? The areolae are simple, but just some images are so good that these occlusions, they look like chambers, but they are not actually chambers. So in terms of protoplast, yes, images are available. It's not in my domain. Oliver Skibe is working on that because, in fact, the way it worked, they isolated the samples in Germany, they had them fresh, and they sent me clean material. So I received clean valves. So I don't have access to the living cultures. And that is maybe...
Someone is recording that. Someone is taking pictures of the live material, though?
Yes,
I see Regina nodding her head. Good.
Yeah, they produce amazing pictures, very beautiful pictures of living cells, but I cannot answer this question.
There are also some difference in the purinoids, the numbers of them.
Oh.
But it's always, I mean, it's always difficult to really pinpoint those things. I mean, the sieve-shaped chloroplast, but it gets some wavy at the edges. I think the biggest difference with Cocconeis is when you have large valves, you see a lot of features, like Rosalina was saying, but the smaller they get, the more they look alike. And that is, I mean, not only in the living cell, but also in the valve structures, you see less and less differences. And so it's really difficult to talk about small valves. The best is to talk about good large valves shortly after oxospore formation. I think Rosalina can identify all of them by that time. But the smaller they get, the worse it becomes.
Yeah, that is absolutely true.
Well, thank you.
Oh, yeah, thank you. As we wait for to see if there are any additional questions, I would just like to also make another note from Sarah to Rosalina. If you are eager to share your diagram on how you made your measurements on Cocconeis, we would love to share that through diatoms.org. And I think you already, I think if you could also even provide your suggestions on how to deal with these these Cocconeis in light microscopy, you know, I think your guidelines are very useful. So if after the talk, if you'd like to share that with Sarah and I, we could potentially share that on diatoms.org. Tom, you have a question?
Hi, Rosalina. I enjoyed your presentation. Thank you for also, tackling a very challenging genus, and awesome oxospores. Beautiful pictures. I have a couple questions. One, I noticed that the valvocopulae of the rbcL valve often collapsed around it when it dried. Why is that? I can't see. That can't be like a natural orientation. It's more on the inside. Is that just part of the drying process?
I don't know how to answer this question. This is how I see them. Yeah, you're right. Probably that is a result of the way the samples are processed. But in fact, what I see, working on this presentation, I was thinking that I never saw rbcL valve with two valvocopulae on the top of each other. Always I see sternal valve with two valvocopulae on the top of each other, which means also I'm with electron microscopy, I was trying to interpret the profile, because the valvocopulae, I mean, if you make a cross section, they have profile. With electron microscopy, in the way I'm doing the analysis, I'm not able to distinguish correctly the profile of these bands. I think that we need different type of microscopy to be able to see what is the profile, but they have a distinct profile, and I think that this is why they stay connected to the sternal valve.
Okay, and I had another comment to ask you about. There have been a lot of saltwater taxa where people have applied freshwater names to, especially in the placentula group. What's your opinion? Do you believe, are there any Cocconeis, do you think, that can span the salinity border and go into full marine salinities?
That is a great question. What I could say that, maybe Regina could answer much better, but right now I'm working with macroalgae, which is cyanobacterium, and I culture, which is freshwater cyanobacterium. I culture it in increased salinity, and I know that it's perfectly survives salinity up to mesohaline conditions. It's growing, it's okay. I think that we may need to run experiments like that with Cocconeis cultures with increased salinity to be able to answer this question. But I cannot grow Cocconeis. It's really challenging task. Regina?
Currently, we have been looking at brackish water, like in the Baltic Sea. That's where the type of Cocconeis scutellum is. We finally got it into culture. It took forever. It took 15 years to get it into culture. We finally got it. I have to publish it. I'm sorry, I still have to publish it. And but parallel to that Cocconeis scutellum, we also found at that time Cocconeis eucalypta, that we called eucalypta, which is looks like, I mean, like a typical eucalypta or like an ectoe, similar to that. So, but it's not the same species. So I think this is the biggest thing. I think they also live in brackish and also in salt water, but they're not the same species. So whenever I see a paper that says we have looked at the stretch of population from here to there, and they have changed, I always go by, oh, how do you know it's not a different species? So I think in that sense, the molecular data helps us there to understand what we're looking at.
Yes, of course.
Huge. It's a huge field to research.
Yeah.
And especially with brackish water. I think the brackish water is very interesting because there are much more different types of brackish water than fresh water. So that's very interesting. I'm sure there are different species there.
Thank you.
Yeah, and I mean, always I think from my point of view, I mean, what the analysis I'm applying, because I do mostly morphology and culturing, But the molecular studies, I think that the great thing that you are doing with this research is that you are providing molecular data for molecular identification of freshwater species. And then if the same genes and sequences they are found in brackish water environments, then you could tell, oh yeah, that species is really growing also in brackish. I mean, this is another way to answer this question with environmental samples.
Yes.
Okay, thank you so much. I'm really fascinated that we had this, discussions after the talk. It took us a lot of efforts to do this study. It's unfunded study. We did it just for the fun because we love this genus. We would like to learn a little bit more. Regine, I hope that you don't mind that I share that information which is in review right now. I didn't ask your permission. And I hope that it's helpful also for the practitioners because I'm coming from this side. I mean, they call this bench taxonomies for years. And we really would like to be able to name the organisms that are working with, right? And I hope that I provide a little bit more practical information also.
Yes.
Yeah, Rosalina, thanks for presenting some tremendous research, and thank you all for having a passion for diatoms and being willing to pour your brains and heart into getting this out there. And I think you bring up a really great point as a person who maybe spans academia and bench taxonomy. I mean, I think there has to be it has to be a two-way street of communication, because I mean, in arguably, not to start anything, but you've probably looked at a lot more diatoms than somebody who might just have named a few. But we're not going to go there. So anyways, I really appreciate you presenting that today. It was very clear. It is very difficult to describe the morphology there. And so it was really nice to see it, and you did a great job illustrating it. So thank you very much. And tune in on, I believe it is February 27th. We'll be talking about long-term records of the Southern Ocean diatoms. It's great to see you all, and have a wonderful day.
Thank you.
Bye-bye.
Thank you. Bye.